Hey guys, how's everybody doing today? I have my nerd gear headphones on because it's that kind of day. So that's what I got going on. But we're going to go ahead with our patterns of inheritance lectures. So I'm going to get started on the first one. And I will share my screen here. Okay. Get set up. Right. Okay. So let's go over these for your pre-lecture assignment. Fill in the blank. Gotta love fill in the blank. I mean, really, they're a good way to kind of go over everything, right? Okay, so this term refers to the genes an individual has. Hang on, let me get my pen to respond. There we go. This is referring to a person's genotype. Hang on, you know how this goes. Can't be too easy for me. All right. Woo. Okay. So a person's genotype, this is the genes that an individual has. Um, so genes then themselves, since we use this term a lot, genes are segments of DNA on a chromosome that code for a specific trait. Okay. Each gene is located in a specific position on the chromosome, and that position is called a locus. Okay. Um, when it comes to alternate forms of a gene, so if I start by talking about like hair color, okay, so we all know that there's multiple hair colors naturally, and so hair color is, created by a gene, but there's lots of different versions of the hair color gene, okay, and those are called alleles. Okay, so alternate versions of a gene are alleles. In other words, alleles are specifically what the genes code for. So I used hair color. This example uses eye color. Alleles are classified as being either dominant or recessive. So dominant alleles mask or hide the expression of recessive alleles. Okay. So if we had, let's go back and stop for a second. Remember that we have two copies, right, of each chromosome. That was chromosome 1, chromosome 2, etc., right? So each chromosome has a copy of the gene. Mom's does, and so does dad's. One of those could be dominant, and the other could be recessive. They could both be dominant, they could both be recessive. And the combination of dominant and recessive alleles is what determines which trait you end up seeing in your body. So since dominant alleles mask the expression of a recessive allele, that means that you only need one copy of it, one copy of that allele for it to be expressed. But if it's recessive, you have to have two copies of that allele, okay? So if it's a dominant allele, you can have one dominant and another recessive, but you'll still see the dominant trait. If it's a recessive allele like red hair, I have to have two copies of the allele that would code for this in order for it to be shown. Alleles are often designated by letters. The dominant allele is usually assigned a capital letter or an uppercase letter. Okay, and the letter doesn't really matter what's picked, usually just something that's not easy to confuse with its lowercase version, because the recessive allele is given the lowercase. Okay, if both chromosomes carry the dominant allele, the genotype is called Homozygous dominant. Homo meaning same. Okay, so here's our two capital letters, two uppercase letters, showing that this person has two copies of the dominant allele. So they are homozygous dominant. If both chromosomes carry the recessive allele, well, they're going to be homozygous recessive. And again, two lowercase. When we have this mix where we have one chromosome with a dominant allele and the other with the recessive allele, that's called heterozygous. Hetero meaning other. Okay. The physical appearance of a trait is called a phenotype. I can do this. Phenotype. The phenotype is determined by the expression of the alleles that are in the genotype. Okay. So where that you can remember genotype and phenotype. Up here, genotype obviously has the first part of the word gene in it. So genotype is the genes. And the phenotype right here has a PH to go with physical appearance. Okay. The way I remember this and would encourage you to remember this is that the genotype determines the phenotype. So it's that mix, which mix of dominant and recessive alleles you have that determine how you look. And not just how you look, but how all of the different organs and systems in your body function. Okay. Let's go ahead and talk about some of these basics. So inheritance. I've shown you an image of a karyotype before, so I just want to kind of go over that again. Again, you've got 23 sets of chromosomes to equal 46 total chromosomes, right? So you can see the 23 sets there. The 23rd set is the XY or XX chromosome, the sex chromosomes. So if you count all these individually, you have 46. In a set, we talked about one chromosome comes from mom, one comes from dad, right? However, those are arranged. So in other words, mathematically, you get 23 from mom and 23 from dad to end up with 46 total chromosomes. For humans, remember that 46 is our diploid number and 23 is our haploid number. All right, so let's talk about alleles and the alternative forms of the same gene. So examples, freckles versus no freckles. I'm trying to go over just some basic Mendelian ones that we'll probably talk about in more depth. Attached earlobes or free earlobes, if you will. Never mind, I think mine are attached. Yep. So if they just come straight in, right there, those are attached earlobes. But if they have that kind of where they hang down a little bit, those are like free hanging earlobes. Okay, so yeah, believe it or not, that's like a trait thing. The other one is about your thumb. If it curves backwards, like mine, hitchhiker's thumb, or if it's straight, which of course I can't, like I'd have to make it do it. Okay, that's another thing. I'll just put hitchhiker's thumb. If I can spell. Okay. So when alleles are mutually exclusive, meaning, and there's an inheritance pattern where, it's just it just comes down basically to what's dominant and what's recessive. Okay. So in this case, a dominant allele, as we have already discussed, is always expressed. Always. Okay. And it masks the recessive allele. So this fact isn't just interesting when it comes down to why you have the traits that you do, but it's actually phenomenally important when it comes to any inherited genetic disorders that you may have, because there's dominant disorders and recessive disorders, which we'll get into more. Okay, so a recessive allele only expressed if there's two copies of the recessive allele. Okay, both copies have to be present or you're not going to see that trait or that disorder. And it's always masked by the dominant allele. Okay? Guys, I'm even on medication at this point. It is taking me forever to get over this. I'm kind of dying. Sorry that you have to hear this every time. Okay, so alleles, mom and dad's copies, right? They're going to segregate during meiosis. If you remember, this is when homologous chromosomes are separated randomly during meiosis. And this is again called independent assortment. And this happens during metaphase 1 of meiosis 1. Why am I telling you this again? Because I want you to be able to start putting what you're learning together, like connect the dots, if you will. So we've talked about mitosis, we talked about meiosis, we talked about how gametes are formed, we talked about genetic diversity. And so now I want you to be able to put all of that together when we're talking about inheritance and what offspring actually exhibit as their traits, okay? And that it's a process of meiosis. Okay. All right. So let's talk about then, in addition to independent assortment, random fertilization. Did I talk about this with you guys? I did. I already talked about this a little bit, but I talked about each male and each female can create 8 million different unique genotypes, or genetic mixes in their gametes, and you put those two together and you get 64 trillion potential unique offspring between a male and a female. Um, then I talked to you about the fact that if you'd been conceived at a different time, you wouldn't look like you as you are right now. You had a 1 in 64 trillion chance of being exactly as you are right now. And that has to do with this process of random fertilization. Again, because which sperm fertilizes which egg is completely random. Each sperm and each egg have a unique genetic combination because of independent assortment. And each fertilization event creates a new being with a unique genetic makeup. In other words, every time fertilization takes place, you have a different egg and a different sperm meeting. So all of this actually, I don't know why I left that out, answers this question about why your genotype is slightly different from your siblings. When a DNA study is done, it will be very apparent in your DNA that you are siblings. But as for why your siblings have slightly different traits, this is why. Um, my sister and I, there's only two of us, and we look phenomenally similar, at least according to people that are meeting us for the first time, and we sound the same. Um, she and I had our ancestry done, and we know on paper our ancestry. We're Norwegian and English. Like all of the white countries, we're from all of the white places. Like as white and colorless as you can get, we're from all of those places. Um, so anyway, what was interesting is that my DNA came back with a larger percent Norwegian than hers did, despite the fact that obviously we came from the exact same parents. So that is just one small example of how independent assortment and random fertilization can affect which genes you end up with. You'd think that we'd have the same um, ratio, if you will, of Norwegian to English genes, but we don't. So there you go. I'm a Viking, is what I say, and my husband's a Portuguese landowner. I don't know why he's a landowner. Yeah, we thought that's the other interesting thing. We thought my husband was Spanish descent from Spain, but he's Portuguese. Go figure. Okay, again, the point of all that random fertilization, independent assortment. Um, okay, we've already talked about this. We went over it in the pre lecture assignment. I'll try to write it. But we have, again, homozygous dominant. I'm not going to write this again, but homozygous recessive, and then heterozygous. Okay. And again, once more, what I wrote in the pre-lecture, this is really important. The genotype determines the phenotype. So we've included a sample here for this bird um, with the sample chromosomes and showing the different color pattern markings that are possible. And then the result is the mix of all of these specific genes determine this bird's phenotype. Some traits, like the ones I listed when it came to like your attached earlobe or a hitchhiker, some freckles, some of those are really the only interaction between a dominant and a recessive allele on a gene. However, many of the genes in our bodies, many of the traits, I should say, in our bodies are determined by the interaction of multiple genes. So things like height, eye color, skin color, hair color, eventual weight, predisposition, those things are all determined by the interaction of multiple genes, multiple recessive, multiple dominant. Um, and so you can see that it gets more and more complex as you think about all the possible combinations of those genes and how they determine what you eventually look like and how your body functions. Try to remember how many I have in this slide. Okay. I am tired of looking at that color. I'm going to do this color. Let's talk about Punnett squares. You guys remember these from high school? So I'm going to switch to my text actually here for a second. So Punnett squares show all the possible outcomes. All the possible outcomes between two different genetic combinations. I want to put that. I'm going to do like that instead. All the possible outcomes for the offspring for a specific trait. So when you're doing a what's called a monohybrid cross, we're just looking at one trait. So if I wanted to know, and I'm oversimplifying, but if I wanted to know what my chances were of having redheaded children, I could use a Punnett square. Again, oversimplifying because hair color is one of those that's controlled by multiple alleles. multiple different genes. Okay. In addition, the Punnett square will allow us to determine things called the genotypic and phenotypic ratios. Yay, ratios. In other words, how many homozygous dominant to homozygous recessive to heterozygotes could potentially be created. Okay, we'll go over this, it'll make a little more sense. Pointing out a monohybrid cross, again, examines the potential outcome of a cross between two individuals for a single gene. Different outcomes represented through the Punnett square, which we then put as genotypic and phenotypic ratios. Okay, so I've given us a Punnett square up here. Let's switch forms so I can fill this out. But I'm going to start with this at the bottom because I've already given us a sample that we can do. Okay. So what is the genotypic and phenotypic ratio for the following? We're going to use freckles, which is actually a dominant trait. So capital F, if there are freckles, the dominant trait, lowercase f if there aren't any. And we're going to cross somebody that's homozygous recessive with someone that's heterozygous. When you have this X, it means you're doing a cross, okay? So somebody that's homozygous recessive is going to look like this, right? Two lowercase alleles. When you fill out a Punnett square, in case you haven't done it before, you put one allele over one column, one over the other. Okay, so this is one individual across the top. By somebody that's heterozygous, that means I'm going to be this if I'm a heterozygote. So now I'm going to do down this side. Okay, so homozygous recessive person looks like this. Two recessive alleles. A heterozygous person looks like this. One dominant, one recessive. I'm going to put, in this case, homozygous recessive across the top, and I'm going to put heterozygous along the side. Now I'm going to cross them. So I'm going to take this F from here and combine it with this F from here. Same here. This F with this one. This little f with this one. This F with this one. Normally, regardless of the order that they're put in on the sides or on the top, when you cross them, you are going to put the dominant allele first, and the recessive allele comes after. Okay. So in this situation, what I have here let's look at our genotypic ratios first. So how many homozygous dominant are there? I'm going to put it like this. Well, none. We don't have any homozygous dominant individuals here. How about homozygous recessive? Well, we have two. And how about heterozygous? I have two. Well, we just answered our question. The genotypic ratio is 0 to 2. No homozygous dominant, two homozygous recessive, two heterozygotes. Now our phenotypic ratio is asking us what did these offspring look like? Let's go through our possibilities. So we know we could have FF, big F, little f, two little f's. This person has freckles, two dominant alleles. Guess what? So does this person, because they have the dominant allele. There's only one needed to match the recessive. This person has freckles. Only the homozygous recessive individual will not have them. So using this information, who has freckles here? Well, potentially 2 of the 4 could have freckles, because potentially 2 of the 4 would come out with a dominant allele. The other 2 would come out potentially with the recessive alleles, so our phenotypic ratio is actually 2 to 2. 2 showing the dominant phenotype, 2 showing the recessive phenotype. So 2 with freckles, 2 without freckles. We'll do it like that. Okay. All right, let's do another example. Maybe if I can move my toolbar out of the way here. Let's keep using freckles. but I'm going to erase all of this. No, I'm not going to erase all of it. I'm going to erase some of it. Okay. I'm going to give us a different mix or a different cross. We're still going to use freckles. Let's do against somebody that is homozygous dominant and somebody that is heterozygous. Okay. I'm going to do the same thing where I'm going to go through, I'm going to cross. This is what I mean when I say that even though the lowercase f is listed first in our cross, we still are going to put the dominant allele in the first place. Okay. So if we were doing our genotypic ratio here, we ask ourselves the same question. How many homozygous dominant individuals are there? Well, there's 2. Nifty. How many heterozygous? 2. Nifty again. How many homozygous recessive? None. Okay. Our phenotypic ratio. How many of these individuals have freckles? Well, these guys are dominant, so that's all they can have. These guys have a dominant allele, which masks the recessive, so everybody does. Right? All 4 of them come out with that. So in this case, all the offspring have freckles. Whereas in this case down here, only, I added a syllable, only half have freckles. Okay? Does this make sense? There are practice Punnett square examples and problems that are listed under this module in a separate file. So if you're struggling to understand this comfortably, I would encourage you to go do that. And then as always, reach out to me so that we can go over it together if you're struggling.